Hello everyone, it's Mrs. Sandra here again, and I'm going to be your lecturer, of course, for MCB 204. Welcome to my YouTube channel. Do not fail to subscribe because in this channel, I'll be taking you guys on everything about microbiology, why you should be in love with microbiology, why you should be proud of yourself as a microbiologist. So, what are you waiting for? Hit that subscribe button, please. You know this autocorrect thing is messing with my mouth, but don't worry. Hit the subscribe button. Welcome to my channel. Tag along, and today we'll be talking about something important. We're talking about gram staining. Gram staining. So that's what we'll be discussing about today. Hit that subscribe button and come on board to my class, okay? It's gonna be fun and exciting. Stay tuned. The most important part of this class we'll be talking about today is the gram staining technique. In 1882, a bacteriologist, don't mind me, bacteriologist, a bacteriologist called Christian Graham in 1882, he discovered this. What is the unique thing about this? Some microorganisms, in particular some bacteria, have thick peptidoglycan layer in their cell wall, and they are called gram-positive organisms. While some have thin peptidoglycan layer, peptidoglycan, okay, and these people are called gram-negative bacteria. So now, the presence of this peptidoglycan layer, this is a pictorial view of the cell wall. This is gram-negative, and this is gram-positive. So the presence of this peptidoglycan makes the primary stain, that is the first thing you're trying to apply on their cell wall, to be retained for gram-positive organisms. This peptidoglycan do not allow these things to go off when you decolorize the cell with alcohol. It does not allow the stain to go off. But unlike that for gram-positive organisms, in gram-negative organisms, this thin peptidoglycan layer cannot retain the cell when you use a decolorizer. We're going to be talking about the 4 stages that we do in gram stain so that you understand what I'm talking about. But this is basically the principle of everything we'll be talking about today. This is why we use the gram stain, and this is why we differentiate these organisms into gram-positive and gram-negative organism. Example of a gram-positive organism, you have the Staph aureus. While for gram-negative organism, you have E. coli, you have Klebsiella. Uniquely, these organisms are very significant in causing diseases, not really because of the peptidoglycan for gram-negative organism, but because of the lipopolysaccharides found in their outer membrane. And we'll still get to that as we go on. So now I want to take you guys on the 4 stages that we'll carry out when we want to do a gram stain. This is very important. It's something we need to know on the fingertips. Like, we need to be conversant with this. So let's get to it. All right. We're talking about the 4 steps in gram staining. I don't know if you can hear from the background, my baby's sleeping. Okay, so we're talking about the 4 steps in gram staining, okay? So the first step we're talking about is, well, I could call it fixation. After you've created a smear on your slide, after you created a smear, how do we create a smear? Using a sterile loop. You can just take a little from your colony that you can find in your petri dish, and you just, first you have added sterile water on your slide. Then using your sterile loop, your inoculating loop, you take a little from a colony that you see on your petri dish and you make a smear. Then you need to fix it so that it can stay. Just pass it over the light, then it can stay. That is what we call fixation. But after you've done that, you pass it lightly over a Bunsen burner, not for long. You probably say for like a second or 2 seconds. or probably 3 minutes. But all I know is that you don't need to waste time when doing your fixation. But we know, of course, we're going to be doing the practical aspects of this, but let's just run through the methods. So after that, after the fixation, the next thing we're going to be applying is the crystal violet. This is the first thing. We're going to be adding the crystal violet using a pipette. You're just going to take from, it's a bottle and it's usually purple or dark blue. You take it and you put it over your slide that you've already fixed. That's the organism that you've already fixed on your slide. And after the crystal violet, the next thing we're going to do, we're going to add a mordant that would help allow this stain, this crystal violet stain, to stick to the organism. And this mordant is called Lugos iodine. So that's what we're going to be adding next. Iodine, it's a mordant. It helps fix this stain into the cell wall. So after the addition of this stain, the next thing we're going to be doing is we're going to do the decolorizer. We'll try to decolorize, like we want to know if this particular stain will go off, because when you're working with this, we are working with like a blind person. You don't know if the organism you have is gram positive or gram negative. Sorry, I just looked over there, my baby's sleeping, but sorry about that. So Lugos iodine, after the mordant, next we're going to be doing, we're going to be adding the decolorizer. This is an alcohol. You can use ethanol or anything. So you're going to pass the decolorizer over your slide for a few seconds, like I think it's 60 seconds, like this is a very significant point in this gram staining technique, because if you miss this step, you're going to break the cell wall, and even the gram positive organism that is meant to retain the primary stain would end up losing the stain. So this decolorizing stage is a very important and critical stage in gram staining. After the decolorizer, we are going to be adding the final stain, which is the safranin counter stain. You can call it the counter stain. Usually safranin. So now this is usually red in color, while the crystal violet is purple or dark blue. Yeah. So now after adding the counter stain, then you wash off still. Then after that, you now view under the microscope. The viewing under the microscope, you use your immersion oil. I'm sure the lab technician will take you guys through, but this is the basic step that we do in gram staining. We have the fixation, you have to fix it. Then we have the addition of the crystal violet. Then we need to add Lugos iodine to act as a mordant. Then we have the decolorizer, then we have the counter stain. These are the basic 4 steps that we carry out when doing gram staining. I hope you guys understand my class. I hope you're conversant with it. And I will also want us to always have in mind the reason why we stain. We stain to have a contrast in the cell wall and the background that you're working with. And we also stain to identify, like when you're doing gram stain, you're trying to identify a particular plate. You have a petri dish, you don't know the organism in that plate, so you want to know if this organism is gram positive or gram negative. Now for a gram positive organism, after you've done this, the color is going to remain when you view it under the microscope, it's still going to have this purple or dark blue stain. Why? Even though you added safranin. This is because of the peptidoglycan layer that we talked about earlier. This thick peptidoglycan layer would not allow this crystal violet stain to go off, irrespective of the fact that you counter-stained. Because of this thick peptidoglycan layer, it retains the primary stain. But if you repeat these steps for a gram-negative organism, it's going to lose the crystal violet stain because the peptidoglycan layer is thin and it cannot stay when you decolorize it. So the gram-negative organism is going to retain the counter stain, which is safranin. So our final result when you carry out a gram stain, you're going to be having, let's clean this, the gram-positive organism to be purple, retaining the crystal violet stain, and the gram-negative organism to be red, retaining the safranin stain. Do we understand? Yes. So these are the basic steps that we do in gram staining, and this should be our final results. So thank you guys for listening. I hope you understood my class, and if you have any questions, feel free to drop your questions down, and do not fail to subscribe to my class. I'll be taking you guys on this class in person, but I wanted to do this so I can get yourself acquainted with my class. So I'll see you guys in class to explain more. But if you know you can't attend my class or you have somewhere to be, make sure you watch this video. Okay, thank you guys. Don't fail to subscribe, and bye. See you in class. Bye.